In this video, we'll discuss enzymes further and how they're regulated. Let's start with a cartoon version of an enzyme. Here's our enzyme, which remember is a protein, and it has an active site, which is where the substrate will bind. And here's our substrate, again just a cartoon version. This represents some type of molecule. Remember that enzymes are specific. This enzyme won't bind just any molecule, only a specific type. The substrate and enzyme bump into each other in the cell and binding occurs at the active site. Now remember, enzymes and substrates probably aren't like a lock and key. Everything in the cell is in motion all the time, and that includes the molecules, which are slightly changing shape and flexing around their bonds. So the enzyme and the substrate bind to each other, and this causes an induced fit where we have just little shape changes to make that binding a little bit closer. Okay, now that we've bound via induced fit, the substrate undergoes the reaction, which is catalyzed by the enzyme. And once it's been changed, we have a product, or perhaps more than one product. In this case, we had a substrate that was broken into two products. Now, if we're breaking bonds and breaking a larger molecule into smaller molecules, what kind of reaction do we have? Is it anabolic or is it catabolic? Hopefully you said catabolic. Now, once our reaction is finished, the products are released from the enzyme, and the enzyme is now able to do the exact same thing again with another molecule of substrate. Now, let's talk about what affects enzyme activity. We'll cover three main categories. First, physical or environmental factors. Second, cofactors and coenzymes. And third, enzyme inhibitors. Okay, we'll start with physical factors. And let's start out with temperature. All enzymes have a range of temperature over which they function. And this, of course, is going to relate to evolution and homeostasis. Let's take a look at a Greenland shark, for instance. This animal is, as far as we know, the longest-lived vertebrate on Earth, and it lives deep in the ocean in high northern latitudes where the ocean water is really very cold, so its enzymes might function at a relatively low temperature. However, if you take an insect that lives in the fynbos of southern Africa, temperatures fluctuate pretty dramatically in this habitat, and you're going to have enzymes that function over a range of temperatures. And if you're a Pompeii worm that lives at the bottom of the ocean near a very hot hydrothermal vent, you'll need enzymes that work at a high temperature. These worms can survive temperatures up to 80 degrees Celsius or 176 degrees Fahrenheit. And we need enzymes to be functional to maintain all of the metabolic pathways in the cells of the organism. That's why homeostasis is so important. Let's look more closely at the temperature profile of an enzyme. We have temperature on the x-axis and rate of reaction as well as percent active enzyme on the y-axis. We're going to concentrate on the shaded curve here, so we're looking at rate of reaction on the y-axis to the left. So the enzyme here functions optimally at about 48 degrees, as indicated by the dotted line labeled optimal rate. Colder than that, in the part of the curve shaded blue, the enzyme works less well. And hotter than that, which is the part of the curve shaded red, we see the activity falling off really quickly once the enzyme denatures. In terms of structure and function, what's going on? Well, if it's too cold, then the enzyme probably isn't going to lose its three-dimensional shape. In other words, its tertiary structure, but it will become more rigid. Keep in mind that one way to think about temperature is as a measure of particle movement. If there's less heat, there's less molecular motion, and the consequence is that the enzyme is less able to bind substrate. And if it can bind the substrate, slower at converting the substrate into product if it does bind. All right, what about the other side of the scale? What if it's too hot? Molecular motion speeds up, and this is going to affect the shape of the enzyme. Over a certain range, increasing the temperature will increase enzyme activity. But if it gets too hot, the enzyme is going to change shape. It's going to be unable to bind substrate, and if it really gets too hot, the enzyme will denature, or in other words, unfold. And it's definitely not going to work now. This has physiological relevance. You've probably heard about people, for instance, that are fished out of frozen lakes or streams. They appear to be dead, no heartbeat, no breath, but you warm them up and they revive. However, if you have a high fever, maybe only, 7 to 10 degrees higher than your normal physiological temperature, that in fact is life-threatening. And part of the reason why we have sort of more room to run on the cold side of the scale is due to what I just explained about enzymes. If you get too hot, enzymes denature, they can't catalyze reactions, and we need to be performing these cellular metabolic reactions all the time in order to live. But if you're cold, you could still be warmed up and recover. In fact, for some surgeries, surgeons will actually cool patients down to the point where the heart stops. Like for instance, if you need to work on the heart, then it might be advantageous for that heart not to be moving at a rapid rate of speed. So you can cool them down and then warm them back up again. And there's even a medical saying about this, which is, you're not dead until you're warm and dead. Because if you're cold and dead, well, you might be able to be revived. Okay, in terms of physical factors, we can also think about pH. So if the hydrogen ion concentration is too high or too low, this can affect enzyme shape as well. And in extreme cases, it could even denature the enzyme. Even if the enzyme doesn't denature, too many or too few hydrogen ions can affect the shape or affect the chemical properties of the active site, making it unlikely to bind substrate and or unlikely to catalyze the reaction. Okay. Let's move on to our second factor, another way to affect enzyme function. This is the presence of cofactors and coenzymes. Cofactors and coenzymes are molecules or ions that an enzyme needs in order to function correctly. They can be inorganic, such as metal ions, for example, magnesium, nickel, iron, manganese, and zinc are all necessary cofactors for certain enzymes. These helper molecules can also be organic, and we're going to call these organic molecules coenzymes. For example, NAD+, biotin, ascorbic acid or vitamin C, and thiamine pyrophosphate. These coenzymes tend to be vitamins or derivatives of vitamins. So hey, you ever seen a food product advertised that it contained vitamins and minerals? Well, this is what we're talking about. And this is a major reason that you need to eat certain vitamins and minerals to allow your enzymes to function correctly. Let's look at a quick example. Okay, this enzyme is alcohol dehydrogenase. It catalyzes the first step of alcohol metabolism, which means it helps to convert ethanol to acetaldehyde, which is shown in the chemical reaction here. Alcohol dehydrogenase needs zinc in order to function. Each subunit of the protein has 2 zinc atoms, one which is present at the active site, and another present at an allosteric site, and this helps stabilize the shape of the enzyme. The substrate is ethanol, and it binds in the active site, and the chemical reaction to change it into acetaldehyde takes place. And then acetaldehyde is released, and the enzyme can do this again with another molecule of ethanol. Let's look at another example. This is dopamine beta hydroxylase, and it helps convert dopamine into norepinephrine. You've probably heard of these molecules. They function as hormones and neurotransmitters. They have lots of really interesting functions in the body, and you'll probably learn more about them if you take a physiology or neurobiology class. But right now we're going to focus on the enzyme. Dopamine beta hydroxylase needs a cofactor, which is copper, and it also needs a coenzyme, in this case vitamin C. Dopamine binds at the active site, and it's converted into norepinephrine, which is then released. Here are some of the important dietary vitamins, and you get sufficient amounts of these from the food that you eat. We have vitamin A, vitamin B1, B2, B6, folic acid, super important to supplement with folic acid if you happen to be pregnant. A deficiency of folic acid can cause neural tube closure defects. We have vitamin C, vitamin D2, and vitamin E shown here. The body can either use these as coenzymes or sometimes use these to make coenzymes. Okay, let's move on to our third category of factors that affect enzyme activity, and this would be inhibitors, so things that stop or slow down enzyme activity. We'll discuss 2 types of enzyme inhibition, competitive inhibition and non-competitive or allosteric inhibition. We'll start with competitive inhibition. This is where an inhibitor molecule competes for the active site. So here we have an enzyme called succinate dehydrogenase, and it helps convert succinate into fumarate. This is a step in cellular respiration. And it turns out that a molecule called malonate is an inhibitor of this enzyme. What malonate does is bind to the active site, making it impossible for succinate to get in there. Now, some competitive inhibitors are reversible, meaning the inhibitor can also exit the active site so that the regular substrate can bind. In this case, inhibitors will work in a concentration-dependent manner. The more molecules of an inhibitor, the less substrate gets converted to product, because the active sites of all of that type of enzyme are occupied with inhibitor more of the time. Some competitive inhibitors are irreversible because they bind so tightly to the active site that they don't leave, or because they change the enzyme somehow such that it is non-functional. Some drugs and toxins are irreversible inhibitors, for example. You're probably familiar with the idea of cyanide and how it's lethal. Well, it turns out that its lethality is because it competitively inhibits cytochrome oxidase C, which is a totally essential enzyme for making ATP, which of course cells absolutely need to stay alive. Let's move on to the second type of inhibition, allosteric inhibition. This is a type of inhibition in which the inhibitor doesn't bind to the active site. It binds somewhere else, and this changes the shape of the protein, including the conformation of the active site. So now our substrate isn't able to bind to the enzyme, and the enzyme isn't going to be functional as long as this inhibitor is bound. Again, this can be reversible or irreversible depending on what type of molecule that we're talking about. This is something that cells use to their advantage in metabolic pathways. A lot of things that the cell makes need to be present in a certain amount. You don't want to keep making the same thing without stopping forever and ever, and that's for a couple of important reasons. One, it could be taking energy that you could be using to do other things. And two, you don't want some substances to build up to toxic levels in the cell. So this is one way to regulate that. In this example, we have a substrate that's undergoing 3 reactions in order to make an end product. So this is a metabolic pathway with 3 steps, a pretty short metabolic pathway. Each step is catalyzed by a different enzyme. Enzyme 1 converts the substrate to intermediate A, enzyme 2 converts intermediate A to intermediate B, and enzyme 3 converts intermediate B to the end product. And the end product, it turns out, is an allosteric inhibitor of enzyme 1, which catalyzes the first reaction in this pathway. This is a really elegant way to make sure you're producing the right amount of product, but no more. And this inhibition is reversible and concentration dependent. If you start running out of the end product, well, less of it's going to be bound to enzyme 1 molecules, which will mean that they'll be active and catalyzing that first reaction once again. Okay, hopefully this video has helped you understand 3 factors that affect enzyme activity: physical factors such as temperature and pH, cofactors and coenzymes, and enzyme inhibitors.